So the next form of cell death will be pyroptosis. So pyro means fever, fire or fever. Tosis means falling off. So this fever is actually going to be because of an interleukin 1. This is the only other cell type wherein caspases are activated. So only two types of cell deaths are there which has caspase activation. The first one was apoptosis and the next one is pyroptosis. But still, the caspases which are involved in both of this is going to be different. So again here it is going to be called as a programmed cell death because it is going to involve certain regulated pathways, and this programmed cell death is going to be associated with inflammation. Okay, so why? Because interleukin 1 is being produced, which is going to cause leukocyte recruitment and fever. So this is going to cause inflammation, right? So this is a programmed cell death associated with inflammation. And here the caspases, it is also going to be caspase dependent, but the caspases activated here will be caspase 1, 4, 5 and 11, out of which caspase 1 is very, very important. This caspase 1 is the one which is going to activate this interleukin 1. So let's see about the mechanism of pyroptosis. So there are going to be generally pattern recognition receptors. So these pattern recognition receptors are going to be of many types. So there is a toll-like receptor. This TLRs are toll-like receptors present on the cell membrane usually. And other than that, we have this nod-like receptors, NLRs, which are present in the cytoplasm. So these toll-like receptors and nod-like receptors, they actually bind to the bacterial proteins like flagellin. So these bacterial proteins are going to be present on various bacteria like Shigella and Salmonella. So these flagellin proteins are present. They come and bind to this toll-like receptors and nod-like receptors. So when toll-like receptor is activated, it is going to cause the conversion of pro-caspase 11 into active caspase 11. And this active caspase 11 will then convert the pro-caspases 1, 4 and 5 into their active forms. Okay. Also, when this flagellin is going to come and attach with this nod-like receptor in the cytoplasm, it is going to form a complex called as inflammasome. So this inflammasome complex is activated, and this inflammasome complex is then responsible for the conversion of this caspase 1, 4 and 5 into their active forms again. So this is one pathway which is happening, and there is another pathway which is happening over here. So now that this procaspase 1, 4, 5 and are getting activated, this caspase 1 has two functions. Firstly, this can directly go and cause membrane damage. So membrane damage is there, it is going to cause inflammation. Then this caspase 1 can also convert the interleukin 1 form from its pro form into an active form. So active interleukin 1 is formed. This again is going to cause fever. So interleukin 1 was the most important interleukin causing fever. Along with that, there is going to be leukocyte recruitment. So there is going to be associated inflammation. Here again, so the morphology will be that of a necrosis because membrane is being damaged and inflammation is there. But still, it is a programmed cell death because it involves some pathway. This formation of this inflammasome is there. So this formation of, so this is a programmed cell death. And this programmed cell death involves the caspases like apoptosis, but the caspases are different, which the caspases involved in pyroptosis, caspases 1, 4, 5 and 11. While in apoptosis, it was 8, 9 and 10 and 3, 6 and 7. Okay. So last form of, last before form of cell death will be anoikis. Anoikis literally means without a home. Okay. So normally cells are adhered to each other with the help of cell addition molecules. But when there is loss of cell addition and the cells are becoming separate, lying separate, so in those cells there is going to be a form of cell death called as anoikis. It is basically a form of apoptosis only. Okay. Then we'll see about ferroptosis. As the name suggests, ferro means iron and tosis means falling off. So there is going to be some kind of cell death associated with iron. So to understand this ferroptosis, you need to understand about free radicals. So let me give you a brief recap about free radicals. So free radicals are single unpaired electrons which are present on the outer orbit. So since this is unpaired, it is going to be highly reactive and unstable. So they tend to go and bind with whomsoever is going to donate another electron for them to become stable. So this is an autocatalytic reaction. So when it comes into contact with some other particle, it will break them also and produce further free radical formations. So this is very highly unstable. So let's see about the reaction which happens in this free radical formation. it happens during normal respiration also. So this oxygen is normally oxygen is present, right? So oxygen on that, if there is going to be any toxin or injury or any radiation exposure, or even during oxidative phosphorylation, so what happens is oxygen breaks into superoxide. So this O2 dot is going to be called as superoxide. This is the first free radical which is formed. Then the superoxide is going to be dismutated. So this dismutation is going to be caused by the enzyme called as superoxide dismutase. So does superoxide dismutase. So superoxide dismutase will dismutate this superoxide ion into hydrogen peroxide, which is H2O2. So this H2O2 will then convert into a hydroxyl free radical. So OH dot is this hydroxyl free radical, and this is the most potent free radical. Okay, this reaction of conversion of H2O2 into hydroxyl ion will happen in the presence of either copper or iron. So in the presence of iron, that is the ferric form will get converted, the ferrous form will get converted into the ferric form. So this reaction, that is the H2O2 converting into hydroxyl ion in the presence of this iron and copper, is called as the Fenton's reaction. Okay, this is the very most important reaction, and depending on this only the ferroptosis is going to happen. So the effects of free radicals will be, it can affect the membrane, protons and the nucleus of the cell. So in the membrane, it is going to cause something called as lipid peroxidation. So lipids, phospholipids are present on the cell membrane, right? So these free radicals will go and react with these phospholipids present on the cell membrane and cause the formation of lipid peroxides. So PUFA is polyunsaturated fatty acid present on the cell membrane. Reacting with this hydroxyl ion, it is going to form lipid peroxides, and this lipid peroxides will in turn cause the membrane damage, because lipid peroxides are not normal components, right? So the membrane now will be damaged by this lipid peroxides. Then these free radicals can cause oxidation of the protons. So enzymes, these are protons. Again, there are also structural protons. So all of these are going to undergo oxidation. So oxidation is it is going to degrade the proton. So it will cause either degradation of the proton or misfolding of the proton. So these protons are going to be misfolded, causing it is going to cause endoplasmic reticulum stress. So there is going to be misfolding of the protons. In the nucleus, again, it is going to cause single-stranded and double-stranded DNA breaks. So free radicals can cause DNA breaks, and this will tend to form adducts. So when adducts are formed, eventually it will lead to mutations. So mutations are always problematic. Okay. So free radicals have very detrimental effects on the body. So how are these free radicals normally removed? Because I told these are going to be normally produced in the body as well, right? So normally they do not cause lots of injury, because there is presence of the free radicals removal. So there are presence of this antioxidants which are going to remove these free radicals. So the most important free radicals was the reactive oxygen species. So generally we use the term interchangeably, right? Free radicals and reactive oxygen species. So antioxidants are basically vitamins, enzymes and transport proteins. So in vitamins, we have vitamin A, C, E, which are called as antioxidants. So that is why they ask us to eat lots of fruit, so that antioxidants are being produced which can remove these free radicals. Then in enzymes, we have three enzymes. Okay, so firstly the same reaction I'm going to write again. So this superoxide which was formed is going to dismutate into hydrogen peroxide with the help of superoxide dismutase enzyme. So this superoxide dismutase is the first enzyme which is an antioxidant. Then this hydrogen peroxide will then form into hydroxyl ion. Simultaneously, this H2O2 can also be broken down by an enzyme called as catalase into water and oxygen. So the second antioxidant will be a catalase. Then this hydroxyl ion and also this H2O2 can get converted directly into water with the help of another enzyme called as glutathione peroxidase. So GPX4 is glutathione peroxidase. So superoxide dismutase, catalase and glutathione peroxidase are the three enzymatic antioxidants. So under transport proteins, we have transferrin, ferritin and lactoferrin, which are responsible to transport the iron, while ceruloplasmin is responsible for transporting the copper. So iron and copper I already mentioned when the it will help in the Fenton's reaction, right? So if these protons, transport protons are not there properly, it will result in some free radical injury. So one other thing about this superoxide dismutase is the superoxide dismutase is the most important antioxidant in the brain. Okay, most important antioxidant in the brain will be superoxide dismutase, and there is a mutation of the superoxide dismutase. So there are two forms of superoxide dismutase basically. We have a copper zinc form and also a manganese form. This manganese form is going to be membranous, while this copper zinc form is going to be cytoplasmic. So, if there is a mutation of this copper zinc form of the superoxide dismutase, it is going to lead to a condition in the brain called as amyotrophic lateral sclerosis. Amyotrophic lateral sclerosis is caused due to the superoxide dismutase mutation in the copper zinc form, since this SOD was the most important antioxidant in the brain. Now that you have understood free radicals, let's move on to ferroptosis. So ferroptosis, I already mentioned, it is going to be iron plus falling off. So it is an iron dependent programmed cell death, and this kind of cell death is going to be different from that of a necrosis and apoptosis. The most common organelle involved in ferroptosis will be a mitochondria, because mitochondria is the powerhouse of the cell. So in here only we are oxidative phosphorylation is happening. So lots of free radicals and ATPs are produced during that processes. So since free radicals are associated here, the ferroptosis will also affect usually the mitochondria. And ferroptosis has been implicated in various diseases like neurodegenerative diseases, diabetes mellitus, ischemia reperfusion injuries, and certain cancers as well. So morphologically, biochemically, and pathogenically, everything is different for a ferroptosis compared with the other forms of cell death. Okay. So let's see the morphology. So normally the morphology of necrosis and apoptosis, the cells was swelling or shrunken, or the nucleus had chromatin condensation, right? But here in a ferroptosis, the cell size is going to be normal, the nucleus is going to be normal, the membrane is going to be intact, and there is going to be mitochondrial damage. Since only mitochondria is damaged, you will have to use an electron microscopy to find the mitochondrial damage. So what are the mitochondrial damaged features? So the number of mitochondria will decrease, their size will also decrease, and the number of cristae will decrease. So these are the cristae in the mitochondria, right? So the number of cristae will decrease, and there will be rupture of the mitochondrial membrane. So the mitochondrial membrane is going to get ruptured, and there will be formation of mitochondrial membrane densities. Okay, so these are all features of the ferroptosis, very, very important. So these are upcoming topics, these are other forms of cell death. So remember, necroptosis, pyroptosis, and ferroptosis, you must read it thoroughly. So biochemically, what do you see? Since it is related to this free radical formation in the there is going to be excess free radicals. So that means that it is going to use up all the reduced glutathione. So antioxidants will be used up. So there is going to be decrease in the glutathione peroxidase activity and reduced glutathione. So these are the biochemical changes we see in ferroptosis. So what is the marker of ferroptosis? So it is going to be lipid peroxides. So lipid peroxides were the ones which were formed as a telltale sign of free radical injury, right? So they were formed in the membrane. So these lipid peroxides will form the marker of ferroptosis. So if you see an increased number of lipid peroxides, it means that ferroptosis is also going to happen. So that is how we identify. Then master regulator of ferroptosis. So this glutathione peroxidase 4 enzyme. So this is the master regulator of ferroptosis as well.